this video we will discuss about the relationship between IMPDH2 and PARP1, and what the final conclusion comes out to be is that IMPDH2 prevents PARP1 activation. So in the previous video we have discussed some of the results of the paper where we have come to a conclusion that IMPDH2 regulates this nucleotide in the nucleus, and that in turn affects the DNA damage repair. So in this video the researchers goes more deep. In this figure the researchers has presented results where they come to a conclusion that IMPDH2 prevents PARP activations. So in the panel A, what they did is they check common regulators or common interactors between PARP1 and TOP2A, as both of them are DNA, both of them are this DNA damage repair. Both of them plays a role towards DNA damage repair in the nucleus. And what they have find out is that the common interactors for the PARP1 and TOP2A is this IMPDH2, or the IMPDH2 is the common interactors between PARP1 and TOP2A, and both of them are the master regulators for DNA damage repair. And these results they come up from the open cell source analysis scoring. And once they find out this relationship, they wanted to check whether this relationship are true or not in their actual cell lines. So they did their study on the cancer cell lines and first what they did is they pull up studies of IMPDH2 and then check whether there is a TOP2A and PARP1 presence or not, and they are able to see their presence. And when they did the similar pull-up studies on the nuclear fractions, they find out a further increase in this signals, that is PARP1 and this TOP2A. Now this indicates that there is a interactions between IMPDH2 and TOP2A and PARP1. And thereafter they wanted to check whether there is apoptosis occurs with this adipocyte in the wild type cells with the increase in the adipocyte concentrations, and compare it with the this IMPDH2 knockdown cells. And what they have find out is that in the adipocytes, in the wild type cells, wild type cancer cells, they are able to see low signals for this apoptotic markers, that is cleaved PARP1 and the necrotic PARP1. So when the apoptosis occurs, this PARP1 get cleaved out, and a part of the PARP will remain in the cytosol, so will remain in the nucleus, and other part of the PARP will move into the cytosols, and these are the markers for the apoptotic cells. So in the wild type pancreatic cancer cells, they see less signals for this apoptotic cleaved PARP marker and necrotic cleaved PARP marker, but for the knockout cells they see the high amount of this apoptotic cleaved PARP markers, PRP1 markers, and necrotic cleaved PARP markers. Indicates that the IMPDH2 absence will cause more DNA damage. And when from this they made a hypothesis that the IMPDH2 will somehow controls the nucleotide in the nucleus. So they wanted to check whether it's there is a change in the nucleotides in the nucleus in the wild type and knockout. So they checked it in different cell cycles, and they are able to see that in the knockdown the nucleotide amount are lower. Indicates that IMPDH2 somehow regulates the nucleotide amount in the nucleus. And when they check the percentage dead cells in between wild type and knockout, they see a significant difference, that in the knockdown the percentage cell death is high, and these high is in terms of both early apoptosis and also in the terms of late apoptosis. For the necrosis, they do not see too much difference. And after that they check different markers of the apoptosis in the wild type and knockdown cells. That is, cleaved caspase 3, it is high in the knockout cells, and also this PARP cytosolic intensity as the cytosolic fragment, it is high in the knockout. So this uh, both of these indicates that there is a high DNA damage occurs or apoptosis occurs. The high apoptosis occurs in the knockdown IMPDH2 knockdown cells compared to the wild type cells. And another thing that they have find out is this adiporibose or monoadiporibose or polyadiporibose in the nucleus that will cause this apoptosis, or that will help in the PARP activations. And this is what they have find out is that in the case of knockdown, this poly or monoadiporibose is high, and this will help the PARP activations more, and it will cause the apoptosis thereafter. And the immunofluorescence image are also shown for the adipocytes 10 macromolar, where we can see that high PARP signals are formed compared to the DMSO case. And these things are also quantified in the panel M, where we can see dose dependent increase in the PARP1 spot with these adipocyte concentrations. This indicates all this indicates that PARP1 high activation, so PARP1 will cause more DNA damage or apoptosis of the cells. So from all these results, what they come out to a hypothesis that come out to a conclusions that IMPDH2 will move into the nucleus because of the high DNA damage demands will cause more nucleotide productions, and this IMPDH2 will then interacts with these nucleotides that are accessed in the nucleus, and thereby preventing this PARP1 activations, and thus this apoptosis of the cells will goes down. This is what they come to conclusions from the experimental results that are presented in this figure and the previous experimental results that we have discussed about this paper. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. And if you have any doubt about any of their hypotheses or their final conclusions, you can also comment on this video. That's all for this video. Thank you for your time.